Hello. Hello.
Sir, good
Good
afternoon.
afternoon.
Am I audible?
Am I audible?
Yes, sir, but it's
Yes, sir.
echoing.
Okay. Thank you.
Subrat, sir, you are not audible.
Yeah.
Dr. Subrat, please check your mic and all. No, no. Otherwise, you can use your mobile phone. Sorry, sir, he is joining within 1 or 2
Yeah.
minutes.
No problem. No problem.
Sorry for the inconvenience, sir. I am joined to the director because some error in my laptop and some internet problem is there. Sorry.
Okay, no issues.
Thank you, sir. Let us start this session. Good afternoon, everyone. A warm welcome once again to all of you. As we step into the fourth day of our 5-day faculty development program, that is next generation molecular diagnostic for pathogens and cancer detection. It is a fantastic to be a part of the excited FDB. I'm sure the discussion and insights today will be enriched for everyone involved. It is my pleasure to introduce our eminent invited speaker, Dr. Rahul Modak sir, for today's session. Dr. Modak is an accomplished scientist and researcher, currently serving as an associate professor and associate dean in research at the School of Biotechnology, KIT University, Bhubaneswar. His work has made significant contribution to the fields of microbiology and epigenetics. After completing his PhD in 2009 from the Jawaharlal Nehru Center of Advanced Scientific Research in Bangalore, Dr. Modak went to pursue postdoctoral training under the esteemed Professor Tapas Kundu at the same institution. His thirst for knowledge and innovation led him to further his expertise with another postdoctoral stint at the Institute of Molecular Biology in Mainz, Germany. Dr. Modak's research primarily focused on the epigenetic regulation of host response during bacterial infection. His team has been investigating the role of bacterial deacetylase and acetyltransferase in bacterial physiology and virulence. Through their innovative work, they have uncovered the significance of these enzymes in both bacterial and mammalian hosts. Recently, they discovered that bacterial deacetylase can translocate into host cell during infection, opening new doors for therapeutic research. This ongoing work also involves identifying potential inhibitor of acetyltransferase and deacetylase as novel target for combating bacterial infections. He has contributed significantly to various high-impact research projects funded by prestigious government agencies in India. Currently, he is leading 3 major projects with high weightage, 2 from Department of Biotechnology and 1 from the Bhabha Atomic Research Center, Government of India. In addition to his research, Dr. Modak also holds significant administrative responsibility. He is the convener of the compliance cell, a batch coordinator, and a member of the training and placement cell at KIT. He is a life member of the Society of Biological Chemistry of India, and his contribution to the society, scientific community, and are widely recognized. We are all excited to hear Dr. Modak insights and knowledge today, and I am certainly sure this lecture on molecular diagnostic tools from bench to bedside will provide valuable perspective on the cutting-edge research to design a molecular diagnostic kit and their application in health sciences. We are truly privileged to have with him us. Please join me in welcoming Dr. Rahul Modak, sir. Welcome, sir, for today's
Thank
session.
you.
Over to you, sir.
Thank you, Dr. Subrat, sir, for your kind words.
Thank you,
So,
sir.
uh, so, and that's a very elaborate introduction he has given. So, thank you for that. And so, this topic, although my lab is on infection and epigenetics, but I do not directly work on the diagnostics part. But definitely, if you look at School of Biotechnology, we have multiple aspects. So, what I will share is what are the different tools that are working, that are emerging for all these purposes. Okay. Share. Is the slide visible?
Yes, sir. Yes, sir. It is visible, sir.
Is it moving?
Yes, sir. Moving.
Okay, perfect.
Thank you, sir.
So, thanks to Dr. Behera and the director of MIT School of Biotechnology for inviting me for this session. And faculty development programs are very important to share our knowledge and share some of our learnings with each other so that we can develop into we can have better understanding of the system. So thanks for this invitation, and I hope you will like the session. So what we'll talk about is different molecular diagnostics tools which are emerging for different applications. So as all biotechnologists, we know that we have primarily four classes of biomolecules. We have nucleotides, we have proteins, we have lipids, and we have carbohydrates. And all these biomolecules are quite uh, important, and their biological functions are very, very diverse. But as a student of biology, we understand they have very, very diverse way of functioning and diverse ways of detection. That makes it more exciting to know how different molecules are getting utilized uh, by different application systems. So the first set of molecule we'll talk about is the nucleic acids and DNA, which is like DNA and RNA. So if I look at the classical approach, so far there were not many tools which were earlier developed, which is used for looking at the DNA and RNA. Now DNA and RNA present is it is becoming very, very emerging process where we use different kinds of techniques. Primarily four kinds of techniques we will talk about is PCR, real-time PCR, sequencing, and next-generation sequencing, which are used for detection of these biomolecules. Now, one common thread among all these methods are they are all amplification-based methods. Means the applications we are using that will amplify the copy number of the DNA or of the nucleotide, basically DNA or RNA. So the advantage with that is even if we have a limited amount of sample, we can really have large amount of signal which can be used for different purposes. So this is the common thread for all these methods. And that is why this method has become the first choice for development of different kinds of detection tools. First, we'll start with PCR. I'm definitely sure that all of the audience know what is PCR is about. So, few points I want to highlight. Quite often, when we see that we think that PCR means anything and everything can be amplified without having prior information, which is not true. PCR needs a very specific, you need to have very clear idea about the gene which you are going to amplify, which you are going to study. So whenever you are doing PCR, you have to keep in mind that this is a target gene specific process. You cannot identify an unknown gene using PCR. Then you need to have, so that means when you said it's a target gene specific process, that means you need to have a gene sequence and the known gene sequence, which is nowadays commonly available from different databases. So next question you need to know is what is the purpose you are doing the PCR, because based on your purpose, the process can vary significantly. And then you need to have the right kind of resources, although now these resources has become quite common in most biotechnology departments. And so what are the advantages of the PCR is it is low cost, it is easy to operate, and you can get the result quite quickly. So that is why uh, PCR has been adapted for diverse applications specific, and it is getting as a initially it was a complementary application for microbiology. Now it is gradually replacing many of the microbiology based detection methods by using molecular detection methods. So this is the typically how the PCR products will look like on an agarose gel, and based on the gene you get, based on your applications you get different sizes of products. Now the limitation of PCR is, first of all, you need to prior knowledge of the gene, target gene. You need to have idea how to get the primers, how to design the processes, and you need to have a moderate set of resources, like you need to have PCR machines, other apparatus, -20 freezers for storage, etc. But the challenge with PCR comes when you have to multiply, when you have to do multiple samples, or what you call as the multiplexing. So people tried, however, because of its robustness and because of its ease of operation, people have really adapted this for different biotechnology applications. Now PCR has been further modified into two major processes. The first one is what you call as the multiplexing PCR. So normally when we talk about PCR, we say that in one single tube, we are amplifying one single gene. But in case of in multiplex PCR, we can have multiple variants. Like you can basically target 4 to 6 different genes in one single reaction. And you can analyze the products by at the in a simultaneous detection method. So if you look at the gel at the bottom, you can see there are on the left side of the gel there are single amplifications, which is essentially doing single reaction, and on the right side there are mixed amplicons, and which are now, if you compare the band positions, they are very comparable, because you have used the same set of primers to amplify, but you have used a mixed template, and that is why you are getting multiple bands, and that is where you can do a simultaneous detection of multiple target genes. So multiplex PCR has really emerged as a diagnostic tool, because there are now certain diagnostic kits available to detect the multiple targets from a single sample. And this has a lot of advantages, because many a times, specifically when you are dealing with clinical samples or environmental samples, we have a very limited amount of DNA. So there is not sufficient material to do multiple rounds of PCR. So in those scenarios, a multiplex PCR really helps to identify the genes. But here again, the same principle of PCR and the same limitations applies. You need to have that. You need to know what are the target genes you want to, or the target piece of DNA you want to amplify. Second is the target piece of DNA has to be diverse, not the same as it happens, not the of same size, because then you will not be able to see them as separate bands on the gel. And you cannot do, it is only a qualitative detection, means you cannot compare the load, I mean the initial amount of template DNA which was present. It will be only say that whether the particular template DNA is present or absent, but you cannot do a quantitation, and you cannot use multiplex PCR for novel targets, because you need to have the prior gene information. So the multiplex PCR can be used in the same PCR instrument. Only thing is you have to design the right kind of primers, and you have to optimize the reaction conditions for multiplexing. So this has really helped in developing different diagnostic kits which are commercially available. Another new technology is emerging, which is called LAMP PCR, or linear isothermal loop mediated isothermal amplification of amplification PCR. So if you see what happens is in when we are doing traditional PCR, we need to have a PCR instrument where the temperature will vary very frequently because to perform the PCR reaction, which is still resource intensive. So this technology, the LAMP PCR technology, depends on isothermal amplification of the DNA material. And here what happens, you use a combination of 4 to 6 primers, and by using a specific DNA polymerase you can use, and what it will do is it will amplify first a bigger chunk, then from that bigger template it will amplify a smaller template, which will and because of the primer sequences it forms a loop kind of a structure, and because of this loop kind of structure, then this amplification cycle keep on propagating and you get multiple copies of the template. So basically you are getting an amplified piece of DNA. So the biggest advantage of this method is this is a isothermal process. So you can even use a simple heating block or a simple water bath to do the amplification PCR. Second, another big advantage is once this can be, you do not need to run a gel to run an agarose gel to detect the PCR product. So the process can be designed in a way that it can be a colorimetric detection. Like if you see the image in the bottom, where you can see there is a difference in the same reaction has been done. If the sample is positive, you have a yellowish color. If there is a negative sample, there is a purple color, and if there is a mix, there is contamination or some kind of ambiguous result, then the color is different. So this colorimetric, so the final product can be detected by looking at the colors also, which is makes it very convenient for using in the deploying in the field and doing in a very limited resource setup. You can carry the whole thing on an ice bucket, like all the reagents you can carry. You can set up right in the field. You can just need to have a water bath kind of a setup, which will give you the product. And hence, if you see now, many of the research agencies, even basically uh, they are basically promoting to come up with an innovative detection kits using LAMP PCR, including ICMR. Recently, if you see, ICMR also has. They said that if you have some research ideas where LAMP PCR can be used, that will be funded for specific detection of specific pathogens. So this is where scenarios can go. So basically, basically what has been appreciated about this technology is the direct application right at the field. Like unlike normal PCR where you have to bring the sample to the laboratory for processing, here you can do the processing directly at the field and get the result out of it. The next, so these are basically the two major variants of PCR which are now being used in different applications. The next level of application comes is which are the based on sequencing applications. Now the classical sequencing application for microbes are 16S rRNA sequencing. It's a very well established method. I'm sure the entire audience knows about it, where you can basically look at the 16S rRNA gene sequence, and based on that you can develop the phylogeny, you can identify the genus, you can develop the taxonomy of the microbes, and thus you can analyze complex microbiomes like environmental samples or gut microbiome using 16S rRNA sequencing. But here the limitation is you cannot have a sequence species or subspecies specific in information most of the time. In that scenario, you have to go for the high throughput sequencing methods. Then also people have developed, so because this can be a much cost effective method for the application, so sequencing is routinely used for different purposes. Apart from 16S RNA sequencing, people also do gene specific sequencing. It can be, say you want to study a particular cancer gene like oncogene, whether that there is an oncogenic mutation or not. All these kind of analysis can be done by using gene specific sequencing method, and gene specific sequencing has become quite affordable in the present day scenario, so that it can be really uh, it has really been used as a diagnostic tool. Now, if you go to any cancer hospital, their pathology lab will typically have this kind of facilities where they will look into the gene sequences, or sometimes even they look into the multi-target panels. Like they know that, okay, for a particular cancer, these are the 10 genes we need to look at the sequence. So you can look at the multi-target sequencing also. So similarly, people are also looking at using these sequencing methods for studying genetic disorders. But remember here, as we move from PCR to sequencing, this becomes resource as well as a skill intensive process. So you need to have trained manpower, and you need to have right kind of uh, infrastructure resources if you want to perform sequencing based studies. But the overall the technology, so the initial cost of the process, the establishment cost of the lab is quite high. However, the running cost of the process is not very high, and that is why this has been used. People are using it very extensively for different diagnostic applications. So, but now next comes the whole genome sequencing. All of us know the theory. So you have to have a genomic DNA library. From there, you will do the fragment amplification, what you call as the clonal amplification. Then you have to run the amplified library on the sequencing machine, which is basically massive parallel sequencing, and sequencing happening through sequence by synthesis. And then you have to analyze the data. So looks very, very simplified, but the biggest challenge of the whole process comes when you have to analyze the data at this stage. Because first round of analysis will be will be when you want to convert. Now just imagine you will have like a typical sequencing run, you will get about 10 to 12 GB of data, 10 to 12 gigabytes of data. So that means you need to have that much of resources to sequence the whole sequence, to process that much of data. So either you need to have a high-end computational facility in-house, or you have to use some of the shared resources. But these resources are not available for free. You have to pay for the resources. Now, first step of analysis will be you have to convert the raw data into sequence files. So, and remember now, the sequence files means the reads are typically somewhere around, I mean, the length of the sequence is somewhere around 80 to 150 base pair, or if you are doing for long read, up to 300 base pair. Now, if you take a simple genome of, say, bacterial genome, which is 5 MB, you will have thousands of reads to analyze. So that itself is a very tedious job. Now, from that initial read data, you have to understand, there are ways to check the quality of that read data. Once you are satisfied with your experimental data, the sequence read, then you have to convert it to aligned sequence. Means these fragments you have to align one after the other, so that you basically in silico reconstitute the whole genome of that sample you have used. So that is where there are a lot of tools required, lot of bioinformatics tools will be required, and a lot of statistical analysis are done. So some of these analysis pipelines are available, but you need to know how to use those pipelines, and you need to have good coding skill if you want to analyze the NGS data. And once you align the NGS data, basically you are reconstituting the genome. Once you have reconstituted the genome from your data, then you will identify this is the organism over there, and then you can ask more complex questions. Like you can ask whether what is the organism, definitely, what are the genes present, what are the possible mutations are present in the whole genome. Then you can ask how many copy number of a particular gene is there, so on and so forth. So that is what you call as the tertiary analysis, and that is what is the functional data. To my experience, this analysis part is the most time consuming and most challenging part of the whole process. Although we often tend to think that this experimental data, the data generation is a difficult part, which is not really true, because now this entire data generation process has become very highly automated, in the sense there are specific kits and reagents available. The protocols are very well standardized. So if you follow the protocol, you will get the result. That is not a problem at all. But once you get the data, the raw data, then what kind of questions you want to ask, that is where your scientific understanding and your process, your question will matter. So people generate lots of data, but they really hardly analyze those data. Even if you take the biggest research papers, like if you take a research paper in Cell Press or Nature, they will have a lot of sequence data, but in that paper, if you see, they will only use 5 to 10% of the data. Rest of the data typically remains unanalyzed for that particular study. And those, however, good part is any data which is sequenced is publicly available. So if you have computational skills, if you are interested, you can take the NGS data. There is a database which is available. You can do extensive analysis, and you can really publish very good work, provided you ask for right kind of questions. So what are the different kinds of applications we can do? We typically, sequencing is for whole genome, but you can also use it for targeted areas of the genome. For microbial samples, the advantage here is you can actually have, in one single next generation sequencing run, you can sequence 5-6 different microbes together. So there is a process called barcoding, which can be used to combine multiple microbes in one single run, and then analyze the data and basically distribute the data based on different microbes, which will really save on the cost of the sequencing, but you get, at the same cost, you can get a lot more data which is available. And present day, the sequencing cost has really come down. Like there are companies, very good companies, who are doing the sequencing, normal sequencing analysis in less than 25-30,000 rupees also. Per sample, less than 25-30,000 rupees. So which is reasonably affordable. And they also do certain level of data support, but it still remains, the biggest challenge still remains, how do you do the tertiary analysis, or you analyze the data as per your scientific questions. That is what you have to ask, the right kind of questions as the researcher, as the scientist. And sequencing data, the biggest advantage is it can be a qualitative data, it can be a quantitative data also. Like you can have quantitative or numbers or gene like copy number, and all these detections can be done by sequencing. So obvious question comes, are we really using the sequencing for applications? Yes. So nowadays, again, In cancer biology, there is called cancer panel sequencing, where there are companies who has developed cancer panels. Means they have identified some 50, 60, 80, 100 genes, which is typically upregulated or mutated in a specific cancer sample. So they are sequencing only those genes from the patient sample, and based on those sequences, they are identifying the mutations or gene duplications, etc., which is happening in that particular cancer. How does it help? It helps the clinicians to decide which kind of drug molecule can be which should be given to that particular patient. Now, again, the same question many a times people ask is, isn't it expensive for a patient to afford this? Now, if you look at a cancer therapy treatment, uh, it goes in lakhs. So, and if you so goes in lakhs, and most of the chemotherapy drugs costs a bit, quite a bit. I mean, that's even the second generation one. I'm not talking about the recent ones. Per dose, maybe 25, 30,000 you have to spend. So if you go in a traditional way, so you will see that uh, the doctor will give the medicine in a trial and error basis based on his or her experience. So they do not know which will drug combination will work very well. But if you have the cancer panel sequence of your tissue sample, then the doctor will have less number of choices, or he has to make less attempts to sequence the gene to give the right kind of medicines based on the sequence information. Now you cannot expect the doctor to give the look at the sequence and decide. It is basically done by biotechnologists or bioinformaticians like us, who basically will look at the entire sequence, look at different parameters, and generate a report which will say, okay, this particular patient sample has these mutations, these gene deletions, etc. So these are the possible drug combinations which should work better in this particular patient. Another application of next generation sequencing has come as the screening of genetic disorders, where all the genetic disorders like Down syndrome, etc., can be easily detected even before birth. So the third application, which is again coming up very extensively, is what you call as the non-invasive prenatal uh, detection, non-invasive prenatal test, or NIPT. Like if you want to have a child at a late age, or if a couple want to have a child at a late age, so there are the chances of genetic mutations and genetic disorders accumulating in the child genome, because the parent's genome will be unstable. So to detect that, now NIPT is uh, routinely recommended by the doctors. Like it will look at 50 to 100 specific genetic modifications and tell that, okay, these are the possible, and they will primarily look at the major genetic modifications, and that will basically tell that whether the child can have some kind of genetic disorders or not. So NIPT is now, and it has it is even more affordable, like maybe around 15,000 rupees per sample. But with the NIPT test, basically you are able to know what is the genetic makeup of the fetus, and then you can decide whether that fetus is healthy to be taken forward and to give birth, or if there is a major genetic disorder, then the doctor, under the legal supervision, can advise what you call as the medical termination of pregnancy. And this is very, very important. Another application of this is also the NIPT is also used by the in vitro fertilization clinics, IVF clinics, because before implanting the embryo, they will ensure the embryo is free of genetic modifications. So NIPT cancer panel and genetic disorder detection panel using NGS has become a very extensive tool and very routinely used tool in present day scenario. I hope I'm on time.
Yes, sir. No problem.
Okay, sure. So then the next application comes when you use the real-time PCR. Theory, again, everybody knows we have two different chemistry, either cyber chemistry, where we use a dye incorporation, or we use a Tachman chemistry, which is where you use a specific probe. Now, when you look at the real-time PCR, so the biggest advantage from regular PCR is this is a quantitative process. So you can either have relative quantitation or absolute quantitation of the template. Means you can easily ask whether you have 10 copies of the template, 50 copies of the template, 100 copies of template, 1000 copies of that particular template. Like if you look at the graph which is shown here, which is a very typical way of doing thing is you have a target template, you basically take a full dilution of the template, but you will see that even with the full dilution, you are able to generate a nice standard curve from which you can quantify the unknown concentration of the template. So this is where the quantitative application of PCR has come in picture, and this is where this has become very popular for different applications. You can use both DNA as well as RNA for as a template, and that there are several applications which are people are using the real-time PCR. The first application is you can look at tissue specific or organ specific expression of genes. You can look at how a particular tissue or microbe, etc., is responding to stress responses. You can, for the disease diagnostic process, you can look at, you can find out gene copy number, you can find out in the gene copy number in the genome, you can find out whether a gene is expressed or not, what is the level of expression, or if you can use even for looking at multiple genes which can be differentially operating in a cancer tissue compared to the normal tissue. In the drug discovery also, this basically real-time PCR is used to look at the gene expression and studying the cellular metabolism or functions. It can be used for pathogen detection in the sample, and thanks to COVID, all of us have now know what is everybody, even the general population who are non-biotechnologists, they also know, okay, real-time PCR or RT-PCR analysis. The term RT-PCR has become a household name, although even for the non-biological people. So this is, and this RT-PCR is become very handy tool, and now it has become very affordable. Like if you see the actual cost of running an RT-PCR, if you use cyber chemistry, maybe 50 rupees per sample. And if you are doing Tachman probe, where you can be maybe 100 rupees per sample, not more than that. And if you are using Tachman probe, it is possible to do multiplexing. That means you can use multiple targets. So your cost for the RT-PCR also comes down. So, but initial instrument cost is higher compared to normal PCR instrument. Present day, a real-time PCR instrument costs somewhere between 10 lakhs to 15 lakhs, depending on the configuration you are looking at. And but running cost is really cheap, and good part is even the analysis is very, I mean, it is much simpler. You don't have to have a lot of statistical tool or computational tool. The data can be analyzed on a normal computer or a tab also. It is very easy to analyze. So that is where it has become so popular and so versatile for different applications. Now you must be knowing that every district hospital or district microbiology facility is also having a real-time PCR machine. Now it's a different question that how much it is being utilized. That is a different question. That is more to do with logistics, but practically it is doable. Then this is also being used to look at the food testing laboratories, animal breeding and plant breeding applications, environmental surveillance, microbiome studies, etc., etc. And the extended application of real-time PCR is what you call as the RNA-seq, where you are using the next generation sequencing to look at the overall genome, I mean genome-wide gene expression studies, so total RNA expression studies, which is again a very, very important tool specifically for the researchers. May not be for diagnostic tools at this point of time, but definitely for researchers. Few of the other technologies which is become quite useful in modern day diagnostics, like if you look at cell-based or chromosome-based diagnostics, we have the classical methods like immunohistochemistry and chromosomal tests. But now gradually the chromosomal test, just looking at the chromosome morphology, it has now been getting replaced by a technology which is called fluorescent in situ hybridization, where you use, basically this is a hybrid of classical chromosomal studies with southern blot, but in this case southern blot is modified in a way that you can do it inside when the chromosome or the chromosome is present inside the cell also. Like you can see over here, you can see the cellular nucleus, and you can find out these green and red dots are basically fluorescent signal. So the probes, like the southern blot have probes, similar probes are used here, and these probes will go and bind to the specific chromosome. So you can find out for a particular gene how many copies are there, whether there is a translocation event, whether there is a, like here you can see multiple copies, here also multiple copies, this is the normal one, here there is a breakage, and so on and so forth. So fluorescent in situ hybridization has now been used again in the cancer diagnostic applications, because we all know that in cancer one of the major thing that happens is there is a translocation of the DNA of the chromosomal fragments from one chromosome to the other chromosome, and this can be easily detected by fluorescent in situ hybridization. Similarly, we can use FACS, that is fluorescence assisted cell sorting, and where you can look at the cell morphology, you can look at the presence of different cell surface markers or intracellular markers, and based on that you can use it for diagnostic. You can use it for diagnostic applications, and the advantage here is FACS can be partly automated. You need less amount of individual, because the analysis is such that it can be done in an automated manner. You do not need lot of expertise of the person who is doing, so it can be it is adapted for in the diagnostic laboratories. So although I'm not saying that classical methods are trashed, but these are the complementary methods which is giving us much more amount of information, and this is where the diagnostics is new generation diagnostics is going. So coming to protein analysis, the classical method says, okay, we can do IHC, histochemistry, ELISA, or looking at the biochemical assays. Like all the pathology labs, they basically rely on these technologies. Either they look at the presence of the protein or they look at the activity of the enzymes. Now this has further been complemented by like using HPLC. One of the biggest success story of HPLC in diagnostics is looking at the glycosylated hemoglobin, or what we call as the HbA1c. So just to give you a brief, so in case of so our hemoglobin gets glycosylated, and the level of glycosylation is proportionate to the amount of blood sugar or level. So if a diabetic patient, uncontrolled diabetes, will have more level amount of glucose in the blood circulation, and hence it will result into higher levels of glycosylated hemoglobin. So if you look at the HPLC profile over on the right side over here, the major peak corresponds to normal hemoglobin, and the peak which is shaded is with blue, that is the one which is corresponding to glycosylated hemoglobin. Now looking at this particular peak, looking at the area under the peak, you can calculate what is the level of glycosylated hemoglobin in the sample, and that is where what gives you the more reliable and measurement of the blood glucose level in a patient sample. So this has now become very routine test in any pathology laboratories, glycosylated hemoglobin, where basically it is HPLC based analysis is done. And for the discovery level analysis, obviously we can go for LCMS, where we can take this, we can identify the novel proteins or protein modifications by looking at the mass spectrum of that particular of the peptide fragments. However, this is very complex. These are very complex experiments. LCMS is much more complex experiments. Now, one point again, I just want to highlight, because this is often we see the challenges. Any protein-based analysis, we really do not amplify the signal to a great extent. So you need to have a substantial amount of starting material. That means you need to have substantial amount of tissue sample, etc. if you want to do any kind of protein-based or lipid-based or carbohydrate-based analysis. Unlike unlike DNA, RNA, where the technologies are basically amplifying the signal, here signal amplification is very minimal. So unless you have sufficient amount of static material, you will not get the right kind of result. So you need to be careful if you want to do this kind of studies. Now, now when we look at the lipid and carbohydrate analysis, it is the classical methods basically rely on biochemical tests, which is very tedious and time expertise intensive. And what is being adapted is using gas chromatography, HPLC, and mass spectrometric based analysis. Like you can see, people, this is how the lipid profile and it will look like on a gas chromatography instrument. So you can see, like, these are the different lipids. Even the unsaturated lipid, everything can be separated out on a GC column, on a gas chromatography system, and identified. So this is now used by food testing laboratories. The metabolite analysis, like lipid analysis and other metabolite analysis, are being used by food testing laboratories to check the quality of the food, to even characterize the food, like look at the antibiotic contamination, look at other toxin contaminations, etc. Similarly, for carbohydrate analysis, we have to rely on HPLC. This is also being used again for food testing applications. However, in the healthcare industry also, HPLC is getting incorporated quite a bit, because now people are looking at the different small molecules which is present in our serum, and based on the small molecules, people are trying to identify different diseases. Like those kind of processes are now being developed. They are being tested for populations, and this is where the new generation diagnostics is moving forward. So I think I will, because I know this, each of these topics can be discussed in very large details, but which is not definitely not possible in 1 hour time. And so I thought I will give you the highlights where the different directions are moving, and what are the different applications where we are looking at, and how as a biotechnologist we can grow, we can mentor our students or look at different opportunities. And now, before I conclude, I will tell little bit of 2-3 minutes about our School of Biotechnology, focusing on the research. So these are the different research domains we have, starting from normal microbiology to disease biology to cancer to structural biology or food technology, bioinformatics. We have a diverse array of research interests of our faculty members, and these are glimpses of some of our facilities, in-house facilities. Like you can see HPLC, you can see FAT, real-time PCR, we have multiple of them. This is our cell culture facility, which is class 10,000, means you can even generate cell lines, etc. This is zebrafish facility. This is our animal house. These are confocal microscope, SEM, etc., etc. Fluorescent microscopes. Then this is our greenhouse, so on and so forth. And already few of you, we are collaborating informally, and we are open for collaboration. Some of these facilities are used, can be shared based on the nominal charges. On top of that, if you look at KIT University facilities, we have the mass spectrometer, LCMS, LCMS, then fluorimeter. These facilities are also there at our central research facility, which can be used by anyone on a payment basis. So feel free to reach out to us. We will try to help each other, help you in doing and getting your experiments done, getting your results analyzed, so on and so forth. Last part, this is the research interest, which already Dr. Subrat has. This is my lab research interest. So where we are looking at, my focus is on epigenetics primarily. We look at the protein acetylation, deacetylation, and phosphorylation processes. We look at the protein modifications in bacteria. We look at protein modification in the host cell. And the bigger question we are asking is how the host pathogen interaction, how the bacterial interaction with the host cells alters the epigenetic profile of the bacteria as well as the host. And so far from my laboratory, we have reported a couple of new acetyl transferases and deacetylases. Primarily, we are focusing on deacetylases right now. So we use a combination of molecular biology like gene cloning, then recombinant protein expression, protein characterization, then studies to initially identify the proteins and enzymes, do the detailed characterization. We do biophysical experiments. Then we also use, I have developed the cell culture based model as well as mice model to do infection studies looking at the bacterial infection. We have separate room for doing bacterial infection. We also have the capability to do gene knockout studies in bacteria, certain bacteria, not all bacteria, but definitely. Then we real-time PCR-based analysis or RNA-seq analysis is part of our studies, and trying to address the epigenetic regulation of the host-pathogen interactions. So these are some of the success stories from School of Biotechnology. many of the companies you must be aware of, like InDNA by Dr. B.N. Banerjee. So Dr. Banerjee was our ex-colleague. He started his company in this department. Once his company stabilized, he's now going very well. So he now moved out from the department and he's completely focusing on his company primarily. Dr. Asim Mishra has developed, so his company is on cancer diagnostics. So he has excellent facility for, and he's giving excellent support to the cancer patient community in the Eastern India by doing all different molecular diagnostics for cancer. Dr. Asim Mishra is into microfluidic based devices. So, and his devices are used for certain analysis of urine as well as blood sample for certain disorders. Dr. Bijay Panikai was a PhD student from School of Biotechnology, and Dr. Mandal is his supervisor. They are into the new generation of protein inhibitors from other sources. His company is doing very well again. Dr. Gopal Purohit, he has founded this Heredity Biosciences. Dr. Gopal was our PhD student some years ago, and he is doing a very good job in training the biotech students. Dr. Vishakha and Dr. Bhavna Gupta, they have their own startup. They are, both of them are acting faculties over here, and they have they are into food technology domain. And Ford Base Care is a company which is really growing fast, not directly related to us, but many of our students are part of their team as a analyst, and they are into cancer diagnostics, the sequencing based cancer diagnostics, and they're really doing well. They are based out of Bangalore. So these are some of the translational stories I wanted to share so that we as faculties can think little differently from our routine jobs. Definitely research, teaching will be there, but if somebody wants to do, these kind of possibilities are always there. And lastly, thank you for your attention, and once again thanks to MIT School of Biotechnology, all the colleagues over there for inviting me. And on the right bottom you can see our department, and these are the different ways to reach out to us. So I will stop here.
Thank you, sir. Thank you so much for your insightful and very relevant topic. Actually, that this is for our FDP. This is very relevant. What you have said that from the molecular research in the molecular technique, how that will be come to the molecular diagnostic. So this is, I think, this is very relevant, and you have already told that how this is used in NIPT, that for the fetus, for the to check the any disorder, genetical disorder, and can in the cancer, for the cancer is already there are some kit established, diagnostic kits, molecular diagnostic kits are established, like a BRCA and
Yes,
a RAS and RAR. These are already established, and
those are very
you have
established,
also
and you can,
mentioned.
yeah, you can also now people are doing extensive panels, 50, 60 genes people are doing together.
Yes, yes,
Yeah.
yes, sir. Yes, sir. So
So
thank you so much, sir, for your talk, and we are expecting some questions from
yes, I
our
think
faculty members.
Moshami has a question.
Yes, sir. She has asked, what is the significance of the liquid biopsy in the cancer diagnosis and monitoring?
So liquid biopsy is primarily for ALL, AML, MLL, like the leukemia kind of cancers, where primarily which is the cancer either, I mean, either of the WBC or the sometimes for the RBCs. So basically, liquid biopsy helps you to figure out how what is which class of WBCs are say infected, I mean altered, and what is the extent of alteration. So people are using, say, not only the regular biopsy test, they're also adding to like a lot of histochemical analysis to understand what kind of changes are there. And further, what you can do is you can take those WBCs, you can even look at the chromosomal aberrations by fish, or you can use the classical chromosomal techniques. But it sounds easy, not easy to do. You need expertise. I mean, you need certain level of training to do that.
I think you have covered that answer for this question. One of the participants from Dr. Soham Prabha Sahu has
Mhm.
raised her hand.
Yeah.
Can we take her? Rashmi Bhera sir, can you take her for the any questions you can ask?
Okay, she wrote.
Otherwise, she can write.
Yeah, she wrote already.
Yes.
How can cutting edge molecular diagnostics tools? How can cutting edge molecular diagnostic tools transform rapid and
Yeah.
precise infection detection from bench to bedside in a daily clinical practice? I will share one of my experience. Some years ago, where there was a collaborative project on mastitis. Mastitis, it's a basically a cattle disease. So basically, it's a pathogen to the mammary tissue of the cows. So what we did, we developed a multiplex PCR-based technology where total 11 different pathogens can be identified by two tube PCR, like two set of PCR, you have to two reactions you have to put. 11 different pathogens can be detected up to the level of species. So how does it help? It helps to identify the species, and then the you can look at the you can decide what kind of antibiotics will be useful for that particular sample, for that particular for the treatment. Similarly, similar approaches, like people are looking at the gut microbiome using this gut microbiome also, and many a times people are using this kind of techniques. Now, when it comes to pathogen detection as such, so far bloodborne pathogen technology is getting developed. It is possible to do. I think I have answered your question.
Yes, sir. I think it's correct for this
Okay.
question, sir.
The next
One
question
another
from
question from
Dr.
Priya Shetty. What are the major
The
challenges and
challenges
limitations
and
of molecular
limitations of
diagnostic?
cancer detection. Uh, you still cancer detection, like molecular detection means if your technology is based on PCR or real-time PCR, that is still low resource intensive, moderate resource intensive. But if you are thinking of doing it on sequence based, like cancer panel sequencing, then you need to have access to sequencing facilities, which goes in crores. I mean, half a crore to 1 crore rupees initial cost. Plus, you need to have a computational facility to support your detections. So, and that takes time. So, one side you need a team who are good in doing the experiments, and other side you need a team who will give the bioinformatics support. For each sample has to be analyzed. So that is where, so it is a teamwork. We have to build up a team for those. So that is why it is typically done by specialized companies like Indian is doing in Bhubaneswar, and like Corebase Care, they're our partners. I mean, academic part industry, we have a good collaboration with them. They are based out of Bangalore, but they have a team of 50, almost 100 people now to support their operations. So you need to have a team if you want to do that. I mean, if you want to go for genomics based technologies, if you want to go for cell culture, I mean, the fish, etc., needs expertise and again facility, imaging facility. So yes, so you need to train your manpower for that. You need trained manpower for these high-end technologies.
Thank you, sir.
Thank
And
you.
another
Uh,
participant, Dr. Bikram Pradhan, raised his hand.
yes.
Can you take, sir? Please, you
Can
write.
you write? Yeah.
Please write in the chat box. Whatever the questions you have, please write in the chat box. Okay, let him to write. I actually. Okay,
Okay.
I think he's getting something error, some error like that. Okay, then sir, can we close this session, sir?
Sure. Sure. So if
So
there is
before
no more
that,
question, yeah, we can close the session.
for that, I want to thank you for one of the thing that you have told to, you are offering for that collaboration. So we are looking for that. If possible,
Definitely.
definitely we'll go for that. We'll do some collaboration for,
Def
and we'll take your expertise.
Definitely.
Thank
So
thank you
that
so much, sir.
thank you so much. Okay. Thank you
So
then.
now, okay, sir.
Okay.
Thank you.
I'll
Now
take your
I'll
leave.
conclude this session. Oh yes, sir. Definitely. Thank you, sir. Thank you, all participants. Okay, we'll meet tomorrow. Thank you.